This is Dr. Ravi Saini. So, in general physiology, the first important concept is about the homeostasis. So, what is homeostasis? So, the concept of homeostasis basically it is the maintenance of internal environment of the body, and basically this concept was given by Claude Bernard, who was the first to introduce the concept of milieu interior. That means internal environment of the body. So basically, homeostasis is defined as the maintenance of the internal environment of the body. So basically, there are some observations which were given by the Claude Bernard. The first one was the volume and composition of fluid are maintained constant. Volume and composition of the fluid, independent of the changes in the environment in which the animal lives. So basically, volume and composition. After that, he suggested the ability to regulate internal environment. The ability of the organism to regulate internal environment is the major reason for, especially for humans, to live a normal life, because external environment is constantly changing. So he noted difference between intracellular and extracellular fluid and proposed that extracellular fluid is the internal environment of the body. So basically, he proposed that the extracellular fluid, the fluid which is present around the cells or between the cells, that extracellular fluid could be considered as the internal environment of the body. Now come to the various factors which are responsible for homeostasis. So there are specific parameters which, on the basis of these parameters, we could say that internal environment of the body is maintained. The first one is an important factor, that is maintenance of pH. pH of ECA is maintained, which could be considered as acid-base balance. The second one is the regulation of temperature. Maintenance of water and electrolyte. So there should be balance between water and electrolyte. Supply of nutrients, oxygen, enzymes, and hormones. Removal of the metabolic and other waste products. So these are some factors which are help, or these are the factors which, on the basis of these factors, we could say that internal environment of the body is maintained. The next important part in this homeostasis is the various feedback mechanisms through which through which internal environment of the body could be maintained. So basically, homeostasis is maintained by these feedback mechanisms. In this flowchart, we can easily see that stimulus or stress which is causing disturbance, that disturbance is responsible for change in various factors which we studied in previous slide. When there will be change in these factors, suddenly feedback mechanisms would be activated. So there is activation of feedback mechanism, and after that there will be effects or responses. It could be inhibition or stimulation of variables. If variable is increasing, then feedback mechanism would bring them down, and vice versa. If factors are decreasing, then feedback mechanism would be responsible for increasing of these variables or factors. And finally, there will be return to homeostasis, or the homeostasis will be maintained. If these feedback mechanisms are failed, or due to some reasons these mechanisms are not able to function properly, then finally that would lead to the dysfunction. Now come to the positive feedback mechanism. So basically, positive feedback mechanism is a process in which the end products of an action causes more of that action to occur in a feedback loop. This amplifies the original action. So basically, there is an action which is responsible for some products. So finally, these products would cause increasing or stimulating these actions. So there is a loop, and this loop is positive feedback mechanism, could be considered as positive feedback mechanism. The common example is the parturition reflex, that is basically initiated by the oxytocin. So basically, when the pregnancy is at the end, the head of the matured fetus presses on the uterine cervix. The cervical distension, there is a distension, sends signal to the posterior pituitary release. When the matured fetus is pressing on the uterine cervix, there is a distension, and due to this distension, signals are reaching to posterior pituitary, finally, which causes release of oxytocin. Further, oxytocin increases the excitability of myometrium and causes further uterine contraction. So basically, uterine contractions are causing causing increased release of oxytocin, and finally, increased oxytocin release would result into the increase in uterine contractions. So there is a positive feedback loop or positive feedback system, and these contractions of uterus further pushes the fetus onto the cervix. Cervical distension further increases the oxytocin, and this cycle continues till the baby is delivered. So this one is a positive feedback mechanism. Here I am revising again that when the matured fetus presses on the uterine cavity, that would result into the cervical distension, and these distensions will send signal to the posterior pituitary. Finally, there will be a release of oxytocin, and this increased oxytocin again causes cervical distension. So there will be a positive feedback loop, and this will continue till the baby is delivered. Now come to the negative feedback mechanism. So basically, in human body, majorly mechanisms or the systems of the body are controlled by negative feedback system. If the activity of a particular system is increased or decreased, a control system initiates a negative feedback, which consists of a series of changes that return the activity towards normal. The common example for negative feedback is the blood pressure regulation. So in this, when the blood pressure suddenly rises or lowers, it initiates a series of actions. Basically, negative feedback mechanisms that will try to bring the blood pressure to normal levels. If there is increase in blood pressure, then feedback mechanisms, especially negative feedback mechanisms, would cause lowering of the BP. But if blood pressure is decreased, so various mechanisms will try to bring the blood pressure at normal level by increasing it. The another example of negative feedback is the regulation of hormone secretion. So blood pressure regulation and the regulation of hormone secretion are the common examples of negative feedback. In case of positive feedback, the parturition reflex is the commonly given for positive feedback mechanisms. The next topic in case of general physiology is the transport of substances through the cell membrane. So here is a whole exhaustive list of feedback transport mechanisms. The first one is the passive transport, active transport, vesicular transports, and transport across epithelia. So these are the various types of transport processes. In case of passive transport, we have simple diffusion, facilitated diffusion, osmosis, filtration, bulk flow, solvent drag. In case of active transport, the common examples are the primary and secondary active transports. In vesicular transports, we have exocytosis or endocytosis. And Finally, there is transcellular or paracellular transport, which considered under the transport across epithelia. So these are the various transport mechanisms. Now come to the simple diffusion. So diffusion is a process of passive transport in which molecule is moving from area of higher concentration to the area of lower concentration. The common example are the diffusion of various gases, urea, ammonia. So here a substance is moving from higher concentration to lower concentration. So that is simply diffusion. No energy is required in this simple diffusion. The second example is the uh, passive diffusion. Here is having a passive diffusion through protein channels or ion channels. So here comes the role of various protein channels. Protein channels are the tubular structures that connect the exterior with the interior of the cell. We have studied that these are the transcellular proteins. These are the bilipid membrane. These are the intrinsic proteins. Basically, they forms protein channels or ion channels. So when the movement is occurring through these ion channels or protein channels, then it could be considered a passive diffusion through protein channels or ion channels. Still, energy is not required in these transport mechanisms. The common examples of protein channels are the basically tubular structures that connect the exterior with the interior of the cells. Various types of protein channels are water channels and ion channels. Water channels could be considered as aquaporins. Here we are having an example of various ion channels. So basically, ion channels are the transmembrane protein with two major properties. Transmembrane means they covers whole width of the membrane. So basically, these are transmembrane proteins. The two common properties are the selective permeability, means that these ion channels are specifically designed for transport of specific ion on the basis of size. One ion channel is responsible for transport of only one ion on the basis of charge and size. The second one is the gating. Gating means ion channels are provided with gate on either side of the channel, and opening or closure of the gates regulates the movement of various ions through them. So we are discussing about the diffusion through protein channels or ion channels. In case of ion channels, basically these are the trans membrane proteins which are having two specific properties. The first one is the selective permeability, means uh, only ion channels are specified for ions for specific ions. The second one is the gating. Gating means these ion channels having gates, and opening or closing of these gates regulates the movement of the ions. Now come to the types of ion channels. So these gate could be the opening or closing of these gates could be uh, resultant due to voltage, due to ligand. Ligand means any specific chemical is attached to that particular ion channels, and there will be opening or closing of these gates. The mechanically gated. Mechanically gated means mechanical stimulus would be responsible for the opening of these gates. So these are the various types of ion channels: voltage gated, ligand gated, and mechanically gated channels. The common examples of voltage gated channels are sodium, potassium, and calcium. These are the voltage gated channels. So basically, membrane potential above a certain threshold value opens or closes the gate of these ion channels. When the voltage potential or membrane potential is reaching above the threshold level due to there is a stimulus. This stimulus is causing increase in membrane potential, and when this membrane potential is more than threshold value, that would result into the opening and closing of these ion channels. So these ion channels could be considered as voltage gated ion channels, because uh, any change, or here we are having increase in voltage. uh, Voltage means membrane potential. There is increase in membrane potential. This increase is responsible for the opening or closing of these gates. Sodium channels. So basically, sodium channels have three separate states. This one is at resting stage, this one is the activated stage, and there is a inactivated state. In resting state, that activation gate is closed. There will be no movement of sodium. When these channels are activated, the activation gate is open and there will be movement of sodium. In case of inactivation state, before this one is the opened stage, closed stage, and this one is a intermittent stage, in which calcium channels are not fully closed, but they are not, but sodium are not able to transport, because activation gate is open, but this inactivation gate is closed. So this stage is called inactivated stage. Here activated gate is open, but still sodium is not fully sodium is not able to transport inside the cell. So in sodium channels, basically, when channel is open, so after opening of these channels, these these channels could not be immediately closed. There will be a intermittent stage in which sodium channels are considered as inactivated. So basically, when the membrane potential becomes less negative than during the resting state, basically less negative means that voltage is rising from minus 90 millivolt towards zero, and finally it is when it reaches between minus 70 to minus 50 millivolt, it causes a sudden conformational change in the activation gates to open. Basically, activation in activation of sodium channels, membrane potential, which is at resting stage, it is around minus 90 millivolt. Due to stimulus, this voltage is going towards zero, means it is going towards uh, it is becoming positive. So when it is reaching from minus 90 to minus 70 or minus 50, then at these voltage levels, minus 70, minus 50, these activation gates will open, and there will be a movement of sodium ions. This is called the activated state. At this voltage minus 70 to minus 50, the permeability of sodium channels, or the permeability of the membrane for sodium channels, is increased up to the 500 to 5000 times. Finally, there is sodium channels are activated, means activation gate are open, sodium channels are moving inside the cell membrane. That would result into the depolarization, which will cause various actions in cardiac muscles, that will cause contraction of muscles. Here comes the inactivation of the sodium channels. As this one is a closed state, this one is the when channel is open, but when after opening of these channels, these sodium channels could not directly go into the closed state. They will go into the inactivated state, and in this inactivated state, this activation gate is open, but inactivation gate is closed. So there will be no net movement of the sodium ions. So in case of inactivation of sodium channels, inner gates closes at the end of the depolarization, and that stops sodium influx. Conformational changes that flips the inactivation gate to the closed state is a slower process than the conformational change that opens the activation gate. So this closing of this inactivation gate is slower process than the opening of the activation gate. So that would result into the uh, that would cause a pause, and after that pause, finally channel would go into the closed state. So at this point, membrane potential begins to recover back toward the resting membrane state, which is repolarization process. So when this channel is in inactivated stage, membrane potential will begin to recover back, and it will bring at the level of resting membrane state or resting membrane level through the repolarization process. Another important characteristic of the sodium channel is the inactivation process. Basically, inactivation gate will not reopen until the membrane potential returns to the original resting membrane level. So, it is usually not possible for the sodium channels to open again without repolarization. So, basically, this inactivated stage is responsible uh, for reaching the resting membrane potential at the original level. When the cell membrane is able to recover its resting membrane potential level, then only it will go into the closed state, and finally which will result into the opening of sodium channels again. Now come to the potassium channels. So in case of potassium channels, basically we have voltage gated or ligand gated channels, and potassium current due to voltage gated channels. These are the various types of potassium channels. In case of voltage gated potassium channels, there are inward rectifying potassium channels, outward transient rectifying potassium channels, outward delayed rectifying potassium channels. In case of ligand gated potassium channels, there are G protein activated, calcium activated, sodium activated, arachidonic acid activated, ATP sensitive potassium channels. So these are the various types of potassium channels under the category of ligand gated. In case of voltage gated, there are inward rectifying, outward transient, and outward delayed rectifying potassium channels. These are the various types of potassium channels. For potassium channels, the gate is basically located on the inner of the channel. The gate closes the cytoplasmic opening of the channel when the cell is at resting membrane potential. basically when the interior becomes less negative and approaches towards the positivity, more and more gates open and potassium efflux occurs. In sodium channel, there we are having activation gates, but in case of potassium channels, the gate is basically located inside the or the inner end of the channel. So when the membrane potential is reaching from less negative to positive, these activation gates are opened and more potassium goes outside the cell. There is an efflux of potassium channels. And this efflux of potassium channel finally results into the repolarization of action potential. In resting stage, when membrane potential it is around minus 90 millivolt, these gates are closed, potassium channels are, there is no net movement of the potassium. And when this membrane potential is reaching from minus 90 millivolt to 0, this voltage change cause conformational change. It would result in the opening of these gates, and finally there will be a movement of potassium outside these channels. That would result into the repolarization process. Basically, potassium channels open just at the same time that sodium channels are begin to close. So the decrease in sodium entry to the cell and simultaneous increase in the potassium exit from the cell combine to speed the repolarization process. So basically, the incoming of the sodium channel is decreasing and outgoing of the potassium channel is increasing, and finally it would result into the repolarization process. When sodium channels are open, there will be an influx of sodium that would cause depolarization. Now come to the resting membrane potential. So basically, resting membrane potential means when the membrane is at the resting level, so there is potential difference across this membrane. What are the various factors which are responsible for this potential? Permeability of the membrane to potassium. So normally potassium is more inside and less outside the cell. Therefore, a concentration gradient exists from potassium from inside to the outside. That facilitates potassium to diffuse out of the cell. So basically, potassium inside the cell, there is potassium which is in higher concentration, and outside there is less potassium. So potassium will have always have a tendency to move out of the cell. Permeability of the cell membrane to sodium channels. It is vice versa to potassium. Sodium is basically more outside the cell, because it would always have a tendency to move inside the cell, or the concentration gradient basically is having from outside to inside. So sodium diffuses into the cell. So at rest, the cell membrane is more permeable to potassium than sodium. So at rest, the potassium is moving outside more than the sodium is coming inside. So this potassium efflux, that is more than the sodium influx, finally resulting into exit of more cations from the cells, which creates negativity inside. That is relative negativity. We can say it positive or less positive, because potassium at rest level, potassium is continuously moving outside the cell. This is causing more potassium ions outside the membrane, and here less uh, positivity inside the membrane. And this less positivity could also be called negativity in relative terms. So finally, at resting stage, we are having positivity outside the membrane. Sorry, this is positivity outside the membrane and negativity inside the membrane. This one is the membrane. This is called resting membrane potential. The other examples of ion channels are calcium channels. So calcium pump also functions similar to the sodium pumps. So basically, under the topic of ion channels, we discussed about voltage gated channels. And these voltage gated channels, we discuss about the sodium channel, potassium channel, and calcium channel. Sodium channel was having sodium channel were having uh, activated stage, that is uh, closed state, opened state, or intermittent stage, or inactive stage. Open stage could also be called as activation stage. In sodium channel, we have closed state, activation state, which is the opening of these channels, and inactivated stage, where there is no net movement of the sodium. In potassium channel, we have opening or closing state. So these sodium, potassium, and calcium channels basically are the common examples of voltage gated channels. Here we are having ligand gated or chemically gated channels. So basically, the common examples of these ligand gated channels are the when the ligands or hormone or neurotransmitters, when they attach to these channels, and the attachment of these substances to the channels, specifically hormones or neurotransmitter, is responsible for the opening or closing of these channels. That is called ligand gated or chemically gated channels. The common examples are the nicotinic cholinergic receptor channels, which are present at the post synaptic neuromuscular junction. The second example is the beta adrenergic receptor channels, which are present on the smooth muscle cells. In mechanically gated channels, basically some mechanical factors are responsible for the opening of these channels. The common examples are the pressure receptors or Pacinian corpuscles. So the channels which are present in Pacinian corpuscles are opened by the change in pressure. The receptor cells or hair cells which are present in the organ of corti or vestibular apparatus are the examples of mechanically gated channels. The next heading under the transport is the facilitated diffusion. So basically, when diffusion is facilitated by carrier protein, then this transport is called facilitated diffusion. Here is a role of carrier mediated protein, which is facilitating the diffusion of ions. Examples are the various sugars, the transport of various sugars into the red cells, adipose cells, skeletal or cardiac muscles. Transport of amino acids is also done through the facilitated diffusion. This one is the specific carrier protein which is responsible for the transport of specific substances. Some molecules cannot diffuse through the channels because the diameter of these molecules is larger than the diameter of the channel. This facilitated diffusion is also an example of passive diffusion. Here still energy is not used. Basically, in facilitated diffusion, that carrier protein is causing movement of the uh, specific molecule. The examples are the sugars which move into the red cells, adipose cells, skeletal cells, or cardiac cells, and the transport of amino acids are also the example of facilitated diffusion. The other common examples are the uh, osmosis, filtration, bulk flow, solvent drag. These are also under the category of passive diffusion. Now come to the active transport. That is really, really important. Uh, so basically, energy is utilized here because we are moving substances against the gradient. So for moving any particular substance against the gradient, energy is required. By passive transport process, the composition of intracellular fluid tries or have a tendency to equalize with the extracellular fluid. But that should not happen. So nature is trying to move inequality, because if there will be an inequality in ECF, extracellular fluid or intracellular fluid, this inequality is responsible for the movement of various ions. So this inequality is necessarily required. So this inequality is maintained through the active transport, because ions or various substances are moving against the concentration gradient. So here nature basically maintains inequality of the fluid in respective of intracellular and extracellular compartments by providing special transport mechanisms under the active transport. Here energy is used. The common examples of active transport is the two categories which could be divided active transport. The first one is the primary active transport and the secondary active transport. In case of primary active transport, energy is directly utilizes. So here directly utilizes metabolic energy for the transport of process. The common examples are the ion pumps, various ion pumps and ATPs. They directly utilizes energy. Examples are sodium potassium ATPs, calcium ATPs, hydrogen potassium ATPs, hydrogen ATPs, mitochondrial ATPs, ATP synthetase, CFTR protein, MDR protein. These are the various examples of primary active transport, and these are the specific locations of these. So these ATPs or ion pumps. The common example is the sodium potassium ATPs, or also called as sodium potassium pump. So basically, it is present in all eukaryotic cells. This is an antiport pump. Antiport means both the ions moving in opposite directions. So here potassium is moving into the cell and sodium is moving out of the cell. Sodium out and potassium in. This one is the antiport transport system. And this one is responsible for the high potassium outside and low concentration inside the cell. Importance of electrogenic pump is that it creates the electrical potential across the membrane which is the basic requirement in nerve and muscle fiber for transmitting electrical signals. In previous slides, we studied that potassium is having a tendency to move outside, and sodium is having a tendency to move inside. Why these tendencies are having? Basically, sodium potassium pump, they are using energy is being used by these pumps, and these pumps basically moving sodium and potassium ions against their concentration gradient, because potassium is having a tendency to move outside the cell, and sodium is having a tendency to move inside the cell. But this sodium potassium channel is transferring 3 sodium ions out of the cell and 2 potassium ions inside the cell against the concentration gradient. Here energy is required. In case of secondary active transports, the couple movement of the driving ion is done with the movement of another molecule, that is driven molecule. One is the driving molecule, and the second one is the driven molecule. So usually, sodium is the driver solute for most of these transport for various ions. The energy created by sodium gradient is responsible for the transport of other solutes. Basically, sodium potassium pump mainly generates energy which helps in transport of other molecules. A common example is the sodium glucose co-transport. Basically, sodium potassium pump. It causes movement of 3 sodium ions out of the cell and 2 potassium ion inside the cell. But it uses energy, so it moved these ions against their concentration gradient. The sodium is now having a tendency to move inside the cell, and potassium is having a tendency to move again the out of the cell. So when this gradient is created by the sodium potassium pump, sodium will always try to move inside the cell. When this sodium will move inside the cell, it will also responsible for the movement of another substances that is called secondary active transport. In case of sodium glucose co-transport, this entry of sodium is also responsible for the movement of glucose inside the cell. So basically, sodium glucose co-transport is an example of symport. Both sodium and glucose are moving inside the cell. In case of antiport, the transport is occurring in opposite directions. One ion is if sodium is moving inside the cell, the other ion is moving outside the cell. In antiport, this is the example of sodium glucose co-transport, because the transport carrier protein has 2 binding sites on its exterior side, one for the sodium and one for the glucose. Also, when the concentration of sodium ion is very high on the outside, very low inside, which provides energy for the transport. And when this sodium is moving inside the cell, it is causing movement of glucose inside the cell. It is called sodium glucose co-transport. And why this sodium is having a tendency of moving inside the cell? Because sodium potassium pump or ATPase moved these ions against their concentration gradient. So we can say that by the sodium potassium ATPs. Inequality was created again across the membrane, and this inequality is finally helping in movement of other substances. In case of sodium glucose co-transport, glucose is moving inside the cell. Basically, conformational changes occurs, which allows the sodium movement to the interior cell, and this conformation will not occur until glucose molecule got attached. Basically, this is an protein. When sodium and glucose both are attached, these are the binding sites. When both these substances are attached, there will be conformational change, and due to these conformation changes, these ions or the substances could be transported inside the cell. Until or unless both the substances are not attached, there will be no conformational change in these proteins. The other examples of sodium co-transport is the sodium glucose co-transport, sodium phosphate co-transport, sodium iodide co-transport, sodium potassium chloride co-transport, sodium chloride co-transport, potassium chloride co-transport. All these examples of symport, because both the substances are moving in the same direction. Here is same direction, inside the membrane, inside the membrane, inside the membrane, and inside the membrane. All the examples of symport. These are some other examples. In case of counter transport, exchange or antipode, the driving or the driven or the driving molecule are moving in opposite directions. The common examples are the sodium calcium exchanger. Here, 3 sodium ions is moving inside the cell, and one calcium is moving outside the cell. Calcium is moving outside the cell, sodium is moving inside the cell. 3 sodium and 1 calcium. So these ions are moving in opposite direction. So here we are having counter transport or exchange or antipode. These are some other examples of counter transport. So on the basis of directions, these carrier protein or transport proteins could be divided into unipode, sympode, or antipode carrier proteins. Examples of unipode are the sodium channel, potassium channel, where ion is moving in one direction. In sympode carrier proteins, substances, both the substances, 2 or more substances, are moving in the same direction. Common example is the sodium glucose co-transport. In case of antipode, both the ions or molecules are moving in opposite direction. The common example is the sodium potassium pump or sodium hydrogen exchanger. Here, sodium is moving out of the cell, potassium is moving inside the cell, and in this, sodium moving inside the cell and hydrogen is diffusing out of the cell in sodium hydrogen exchanger. In this slide, we can summarize the whole transport. This one is the example of active transport. Here we are having passive transport. In active transport, there is primary active transport, secondary active transport. In secondary, it could be co-transport or in the same direction, or it could be in the opposite directions, called exchange or antipode. Primary active transport is pump mediated. Sodium potassium pump is a common example. And the secondary active transport is the carrier protein mediated. And passive diffusion could be channel mediated. Here, ions or substances move again according to their concentration gradient. No energy is used in passive diffusion or passive transport. The other examples of transport are the vesicular transport. Here, two important mechanisms are exocytosis and endocytosis. The resting membrane potential, that is really important topic. These are the various factors which are